Hello, everybody. Essentially, we're back for chapter 2 of this morning. I just gave you in the previous talk. I gave you a bit of an introduction to distributional ecology, but that was a very general introduction. It would apply to bears and tigers and mosquitoes and viruses, but it really didn't include much that is particular to or specific to pathogen transmission systems. So now we're going to go a little bit more in detail about how are disease systems different. And so I'm going to turn you over to Luis Escobar in New York, and he will talk with you about this.
Hi, thank you, Tom, and thank to everyone to be here with us again. I would like to share my screen with you. Okay. I hope you can see my screen now. And let's talk about the second part of Tom's talk. And first, I would like to start with the topics. We're going to see now how diseases transmission systems are similar with some systems related to biodiversity, mapping biodiversity versus mapping diseases. And following the previous talk, we are going to see in detail some aspects of interactions among species. This it is the B in the BAM diagram. Then we are going to see some concepts of occurrences in disease mapping. What is the meaning of absences and what is the meaning of presence? That sometimes is a bit different compared to mapping biodiversity. And then the question that we always say in all presentation, what will we model? Okay, let's start. How disease transmission are similar to other areas of ecology. Well, diseases are produced by pathogens, and pathogens are parasites. So let's talk about parasites. Well, parasites are set into groups. There are in the left the macroparasites, microparasites, and in the right the macroparasites. The microparasites usually have replication inside the host. They can have direct transmission or transmission using vectors, and they develop immune response in the host, in the natural host. On the other hand, the macroparasites usually are bigger. They grow in one host, but they can have a reproduction in another host or in the ground. For example, we can have a endoparasite that is going to have the development in one host, and then is going to produce eggs, and is going to release the eggs in the landscape. So they usually have direct transmission and may or may not induce immune response. I'm going to show you some examples of how we can apply concepts and theory from ecology into disease ecology. For example, this is a very robust and useful concept, the R naught, that is used in biodiversity to calculating population growth, but in diseases usually is used in epidemiology to estimate the rate of disease spread. But it's the same concept. In disease ecology, we can talk about pathogens that are specialists, and other could be generalists. So, for example, we have a pathogen that is a specialist, and it is going to look for a balance in the virulence. So, it's going to try to keep the host for a long period. So, it's going to look for the perpetuation, the parasite too. So, for example, the influenza in humans do not have a high virulence usually. On the other hand, we have Generalists that has high virulence in one species, but they are going to have low virulence in another species. For example, plague that has low virulence in some species of rodents, but it could be very, very virulent in another species, including humans, for example. So these kind of interactions are a nice example of coevolution. So I show you here some pictures of bats. I like to work with bats. I collect these pictures from a group of people that is working with bats, and in the left we have these small spots are losses, and according to the researchers that found these bats, they looked apparently healthy. And at the right we have some myiasis, that are these worms that grow inside the skin of the host, and in this case the bat was apparently healthy. But we can have some ticks too. These are ticks found in a vampire bat, and we found in this bat in Guatemala, and the bat was apparently healthy too, with good weight, for example, good behavior. This is interesting because the parasite is not making a lot of damage in the host, right? But let's talk about coevolution with a weird example. Usually, the parasite is not going to try to kill the host, and there is only one evidence in which the specific parasite of one species kill the entire population of the host. This example is from a Microsporidium parasite from one snail, but the population of the snail was reduced by another factors, including habitat loss and pollution. So high levels of Microsporidium drive the host species to the extinction. So, but this is an unusual case. So, because what happens if the parasite kills the host? Well, the parasite dies too, right? And that is known as co-extinction in biology. So another example of coevolution is rabies in bats. And here, what the researchers did is that they inoculate rabies in a group of bats, of vampire bats, capturing into the wild, and they put 100 the doses to kill mice, a group of mice, and the result was that the result was that 75 of the bats survived to the challenge, and this is surprising because no bats were killed by the lowest doses of the rabies inoculation. So this is a nice example of how bats co-evolve with this virus that is known to be the most mortal virus in the world. So another application of ecology into disease ecology is predator-prey relationships. And in fact, a lot of models of diseases are explained using pathogens as predators in the models. So we usually learn in the school that the huge animals are the top predators in the systems. For example, in the Americas, we know that the jaguar is a top predator, and it kills, for example, crocodiles. It kills anaconda, that they are huge snakes. It even kills turtles, for example. So nobody eat them. But we have in this example that maybe one vampire bat can inoculate rabies into the jaguar, and then it is going to produce the disease in the jaguar. The rabies is going to kill the jaguar, and in the species that are in the trophic red are going to have benefits from this interaction. So in this example, the top predator in the system is the pathogen. Another example of how apply ecology into disease ecology is thinking in diseases as predators, because they can control population growth. For example, rabies can kill even 84% of the newborns in one population. But how diseases are different? Well, I'm going to talk a little bit about biodiversity, but you can see the first presentation in this course that talk a lot about this topic. And something that is great in biodiversity is that they have adequate recognition of all the species. There are people that has specializations to identify the species using bones, using genetics, and they have samples stored for a long period under a good, they have a lot of samples that are available for people. So additionally, people from the biodiversity has a lot of specimens from a lot of areas, and they can provide detailed information from each individual, even the location from which it was collected. And there are a lot of robust and freely accessible databases online. And this figure was taken from the book of Town, and you can see that there are a lot of species that dominate the databases, but the most important are birds. In the other hand, we have databases that are made for vectors, for example, and here you can see that the most abundant information are for mosquitoes, but we can a bit of information for lice, for example. So when we compare this group with the vertebrate group, we can see that there are not plenty of information for these species of public health concern. And when we look for databases that are specialized in diseases, I found only this one that provides information from several diseases, but the data is not very clean, and we must invest a lot of time cleaning the data, looking for the reports, and identify the location for each report. So the amount of information from public health compared to biodiversity is very, very poor. When we model biodiversity, usually we think in single species that we can describe carefully in the geography and into the environmental space. That is the case of biodiversity models. But when we think in diseases, the systems are very complex and a lot of actors are involved in the system. Something that is very important in diseases compared with biodiversity is model evaluation. We're going to talk more about it in the future, in the next classes, but if we fail to predict the species presence, maybe the consequences are not going to be very important. I mean, if you do not predict what is the geographic distribution of pandas, the population maybe is not going, the human population maybe is not going to be very affected. But if you fail to predict the disease's presence, maybe you are going to have different consequences in human populations, and that is going to affect your work, right? Let's talk a bit about B in the BAM diagram. And according to the previous talk, interactions among species are particularly important to model disease distributions, because usually we are not going to model single species. The most simple model in diseases include at least two species. For example, the pathogen and the host. And one example of these systems are HIV, that is a virus. But we can have more complex systems, including the pathogen, vectors, and hosts, for example, with malaria. Or we can include a reservoir here in which we have the pathogen, reservoir, and the host, for example, for rabies virus. And we can have the most complex scenario in which we have a pathogen that includes reservoirs and use vectors for transmission to affect terminal hosts like humans, for example, with plague. Diseases are complex because the presence of diseases are influenced by the immunity of the host. So if the host develops immunity, it's not going to be more spread of disease. Another point that is very complex when modeling diseases is that social and cultural factors are affecting the presence or absence of diseases. This is obvious because we are trying to avoid the presence of diseases in one population or in one area. For example, there are some countries that are going to have water scarcity, and on the other hand, we are going to have another area that have a lot of water, and even if we have the same environmental conditions to the presence of the disease, we could have different social and cultural factors that are going to avoid the presence of the disease. So we are not going to have the disease. One example of this is cholera. Cholera is known or was reported in coastal areas of Germany. So we know that there are suitable conditions for cholera in the coast of Germany. But when we check the situation of cholera in the world in last years, we can see that Germany is not reporting cases of cholera. So we can follow the evolution of the current pandemic of cholera, and we can see that cholera was reported in Germany several years ago. So we know that cholera has suitable conditions there, and that there we found cases of cholera in the past. So we made this model of suitability of cholera in seawaters, and we found that Germany has a lot of suitable conditions in the coasts, and the endemic areas of Bangladesh have suitable conditions of cholera too. But when we see the human development index in both areas, we can see that the development level in both countries is very different. So the suitability is present in both countries, but there are social factors that are different in both countries. So, for example, Bangladesh, we know that has several problems in the development and the living condition of the human population there. So that's why we expect that more cases of cholera are going to be present. Additionally, another complexity when modeling diseases is that all the people is working to avoid the presence of the diseases actively. For example, we can have the reduction of the exposure to the pathogen, for example, for HIV or for malaria, or we can provide immunity to the population against virus, for example, Or in the case of malaria, we can have strong efforts to reduce the number of vectors in the system. Or for rabies, we can work to reduce the available reservoirs. Okay, now let's talk a bit about occurrences when mapping diseases. What means absence of species in the biodiversity models? Well, maybe we are not going to have a detection, so that's why we do not have reports of the species, or maybe the species was removed from one area and that's why we do not have reports of the species there, or maybe there are not dispersion capabilities for the species in the area in which we are looking for them. That's M in the BAM diagram. That's the case of biodiversity, the single species models. But when we work with diseases, the meaning of absences of cases is different. For example, we can have the vector and the host, but no the pathogen in the system. So we are not going to have the disease. And even if we have a lot of bites from one vector, we are not going to develop the disease. So that's why the disease is not in the system. Or we can have the pathogen and the host, but not the vector to provide transmission. So we are not going to have the disease in the system. That to show this, I take the example of West Nile virus that includes mosquitoes, birds, horses, and people in the system. So if we remove the vector, we are not going to have transmission in the system. But another example is that if we can have the pathogen, the vector, and the reservoir in the system and the terminal host, but we are not going to have disease. And that's because we are providing immunity to the host. So we have all the components in the system, the pathogen, the vector, but we are not seeing the disease because the population is protected against the pathogen. To exemplify this, I'm going to talk about cholera. This figure is showing the time of infection. Here we can see the first infection, and then these lines are showing the level of immunity provided for the classical strains of cholera. And here is the El-Tor strain of cholera. So you can see that one strain is providing a low immunity, but for a long period, and the other strain of cholera is providing high immunity for a shorter period. And this is happening in natural conditions. So what we see in the field is that we can have a very important outbreak of cholera in one population, and the next year or the next season, we are going to have a more small and smaller outbreak in the same population. And that's because the population acquire natural immunity from the previous infection. So that's a very important issue when modeling diseases that is very different when working with biodiversity. And another example of absences is that we can have the pathogen, the vector, the reservoir, and the host, but there are no diseases. But that's simply because we have a lack of surveillance. And to show this, I'm going to use the rabies in Guatemala, in which usually public health agencies has limited people or technicians working in the field to collect samples. That's known as active surveillance. So what the technicians do is that they collect only a few samples from a broad area just to have a small representation of what is happening. So I remember that when in 2009 we have the outbreak of rabies in North Guatemala, only one brain was sent from each farm, even when a lot of cattle was infected. So that's a underrepresentation of the real situation of what is happening in the system. And what does presence of cases mean? Usually, we are going to have a lack of pathogen in the landscape, no vectors and no reservoirs, so there is no possibilities of transmission in the system, but we are going to have reports of disease. And that happens usually when we are talking about people that travel and acquire the disease in another areas. Or we can have the eradicated pathogen, no vectors, no reservoirs, but we are going to have some reports of diseases. That's weird. What if we do not have the actors in the system and the people do not travel, but we have reports of disease? And we can show this with the Trypanosoma cruzi example and the Chagas disease that includes a vector, the pathogen, and usually a long time of time required to show the infection. So we can have acute disease infection in which we are going to see the symptoms really soon, but we can have another case. For example, here I'm showing a herd in which that is affected by Chagas disease that shows symptoms around 30 years later. So we can have the report of the disease a long time later, even when we remove the actors in the system. So if we are going to model that, we are going to have a lot of noise about what is happening in the area. So what will we model? We can model the vector. For example, if we can work with vectors that are in the land, in the ocean, or need air. We can model the reservoir. We can model susceptible hosts, and to model the hosts, we can work again from species that live in marine environments. We can work with poultry, or we can work with plants of importance for conservation or agriculture, and we can model infected hosts. When we have only data from infected hosts, we can talk about the black box according to Peterson 2007, in which we are modeling maybe the infected host or positive humans cases, and we can have an approach of what is happening in the system, but we do not have the complete picture. But it's a first step to do research and to identify key areas for surveillance, for example. Or in the more complex cases, we can model all the actors in the systems. So I guess that the main advice should be think biologically when you are making these diseases, and when you are modeling diseases, and when you are interpreting your results. So that's all from me, and thank you for listening.
Thank you, Luis. I'm not seeing any questions coming in quite yet. If you have questions, please do send them. For the moment, I think what we'll do is stay on schedule, and so we will be back at 11 a.m. my time, which is 5 p.m. London time, which is 7 p.m. Nairobi time. So please tune back in at 11 a.m. my time, and we will be back with one more talk and then some question and answer. Essentially, today is focusing on the distributional ecology of diseases. And so we want you to have a good, solid conceptual foundation for that before we go on to tomorrow's topics, which are how do you replicate that distributional ecology? How do you measure ecological niches? And how do you estimate distributions of species? So hit us with questions, and we will be back in 29 minutes with the next installment.